University of Algarve. He's going to give a nice talk about interpreting CNN calibrations, and later on we have also the practical session done by Dario.
Yep.
So, thank you.
Okay, thank you everyone for having me here. Just to start with the disclaimer, the fact that I am one of the few people probably in this house working on the interpretability doesn't mean that I know too much about interpretability, okay? So just that disclaimer. Some of the stuff that I'm going to tell applies also to the tutorial we're going to have afterwards. You might have already received a text file with some instructions. So if everything goes okay and the gods of technology help us, it's going to flow. So the idea is to have you guys make some practical stuff in terms of implementation. The code is there. I'll explain the code. The code is in Python, but you can adapt it to Julia, R, or MATLAB, whatever is your weapons of war. So the concept is what I'm going to focus, okay? This session, it's called Interpreting CNN Calibration and slowly developing a deeper understanding. Slowly there should have been put in bold because it's, well, there are several factors contributing to this slowly development. So what does the literature say about CNN applications, specifically in the food industry, since we are AI for food? There are several examples that show that, well, CNN tend to outperform traditional methods like PLS, SVM, SVR, especially in complex spectra, and especially when the way of taking out the spectra is not the finest protocol with your Bruker 5000 in the laboratory. So when you go from the lab with very clean conditions, very high accuracy, resolutions, etc., to the field where you have temperature changes and clouds coming in, and your PhD tilting the spectrometer because he's sleeping, whatever. There's a bunch of conditions that sometimes they messed up the linear models. Preprocessing helps a lot, right? But sometimes they just can't handle it. So there is a bunch of the reviews. You want to have access to the slides. That's why I put all the references here so you can consult yourselves. That show that in very instances, especially in food products and biological samples in general, CNNs tend to perform better than these traditional methods. Now, there are advanced methods like local, like KNN, PLS, etc., that also perform well, okay? But they're not as usual as these ones, so they might be still in development phase, but still work very well as well. So why or how do these CNNs work better? Okay, what do these algorithms have special that work better for a certain spectra? Well, in terms of preprocessing, we have found out that the convolutional layers, and we will see this a little bit front, processes a sort of data-driven optimization preprocessing. Instead of choosing if I'm going to do SNV plus second derivative or MSC plus first derivative plus mean centering, whatever, this optimization that is done on the filters of the convolutional layers tend to find a clever filter that sometimes aggregates these preprocessings into a single filter. Now, we're dealing with 1D vectors, okay? So if you find someone from signal processing, people that work in electronics and stuff like that, they will tell you there's a bunch of 1D filters that are like windows that slide around. So even the derivatives, they're just one filter with some coefficients that slide around and compute relations between data points, and that's a filter, right? So convolutional layers learn this type of 1D filtering windows. In data sets that have many heterogeneities, like batches, sample types, etc., CNNs tend to be more robust because we have found out that CNN do also a sort of variable selection. They tend to find the variables that are more informative, in our case, what wavelengths or what bands are more informative in terms of optimization of that prediction that we want. It can be dry matter or protein content or moisture, whatever, across different samples. And so it tends to use those informative samples or informative wavelengths. Well, there are certain types of peak shifts, for example, related to temperature fluctuations in your spectra that can be absorbed by the convolutional operation or by using pooling layers. So while some methods, like for example PLS, is very sensitive to the shifts in the presence of the peak due to temperature shifts, if instead you have a window that absorbs whatever comes in this wavelength range, and you have a pooling layer, or you have an operation, a convolutional operation that takes care of that interval, then it becomes a little bit more insensitive to, more robust to temperature fluctuations and other types of this type of shifts, like for example, spectrometer from model to model, not different brands, but the same brand, different apparatus. Okay. We will talk about this a little bit later. So in deeper models, we read 3 layers, okay? Everything I'm going to talk, it's up to 3 convolutional layers, 4 maximum, mostly 1, okay? So it's deep learning because it's the paradigm, but it's not deep at all, okay? CNNs learn certain hierarchical abstractions. There's a jargon here from computer science. We will look into it. That by combining non-linear level features, and we will see that, what's happened. And multitask predictions and easier data fusion from different modalities like spectra and images, it's one of the reasons why CNN is getting a little bit boost also in hyperspectral imaging, on where you have, for example, electronic nose information, or you have spectra from MIR or NIR or NMR, and you want to combine them. There's an S missing here, I think. No. The CNNs are able to combine this in a very flawless way, actually. So here are some of the results, that some of the publications that back up these claims. So there's one of the first papers, actually the paper that made me start working on this subject, by Cui and Fern, where Cheng Hao showed that some filters work like the first derivative preprocessing for the spectra. Xiaolei Zhang also shows that some filters work as baseline removal, so they can shift the baselines. In a recent paper, showed that some filters work as like a little bit asymmetric second derivatives, like a combination of baseline shift and second derivative. And there's also other works where deeper architectures were able to, first predict temperature, then use that prediction of temperature to enhance the Brix prediction of some fruit. Now, you have seen this before. I just want to make it for 1D. It's basically what we use. So this is your wavelengths. This is the way I usually like to think. It might be not the best way, but it helped me conceptualize things. So you have your wavelength. This is what we call the input layer. This is where you put your spectrum. Then this is a direct mapping. There's one-to-one connection here. Then you have a convolutional layer, which is represented by 2 operations here. It's the convolutional filter that does the convolution. This filter slides, multiplies each element of the filter. For example, this filter has like 1, 2, 3, 4, 5 boxes. This filter slides, it makes a one-to-one multiplication, then it sums up everything. That's the convolution. And then you have a activation layer. Usually this is this, in practical terms, the activation is within the same convolutional layer, but it's a different mathematical operation that is implemented inside the convolutional layer. Okay? And then usually you have a flattened layer just to put all the dimensions stretch up in a single vector, because here you might have different filters. You can have more than one filter, okay? So you need to straighten this up. And then you usually have one or two or three dense layers where it's easier to think what happens in the dense layer. Every pixel, every unit here, okay, every neuron looks into connections to all these others, okay? And usually there's this one weight associated with each connection. This is a fully connected layer or a dense layer, and this is the type of layer where most parameters come in, okay? So, and then usually you have, for regression purposes, you have one final layer with one neuron that aggregates all the responses, weighted responses from the previous layer, and you make a prediction. Usually here you can have different activation functions. It can be a ReLU, which is something like this and this, or you can have like ELU, which is an exponentially grown up and then linear. Well, it depends a little bit on your choice and optimization. Usually in the final layer for regression you have a linear activation. It's basically x equals y. Okay. Then sometimes we also resort to the so-called pooling layers, which is basically you have the same thing, input spectrum, have the multiple convolutional filters. Each filter goes up here and learns something different from each other. And then from the low-level features represented here, we're just going to we're going to pull whatever we want, which is like the average of this window. We can pull the maximum value of this window. So this usually contributes to dimensionally reduction of the data. And usually following the pooling layer, you align all the channels. So you have a pooling layer for each channel. In this case, we have 3. And we stick them up all together, get 1D vector again, pass it on to a dense layer to non-linearly combine all these features extracted by the convolution and make a prediction. Okay? So the way I see it, building, even if we are just talking about convolutional neural networks, okay, there are many Lego pieces that you can stick up in different ways in order to do your modeling. So you have different filters. You might have filters with different widths. So some can be narrow, some can be wide. Depends if you have any intuition about the spectra you're using. For example, if you're applying a CNN to NIR spectra, which is very smooth usually, you can go for a broader filter. If you're applying that to Raman spectrum, which peaks very, very, very narrow, it doesn't make sense to have a very large, so you can use a narrow one. So there's pooling layers that sometimes help with dimensionality reduction. Personal experience, they screw up the metrics, pooling layers, because when you're going to do explainability, the fact that you use pooling layers means that it's difficult to go beyond that point. I mean, for example, this is one way that I like to think about neural networks. So you have your input vector, right? Your input space, your data. And what each layer does, it's a bunch of matrix transformations. So it squeezes the data, it stretches the data, it folds the data. Sometimes you can fold it really neatly, right? And you have this, okay? But sometimes when you do automatic optimization, stuff like that, this is what we have here. A low-level representation of my data that has been transformed by these non-linear operations in the convolution and all the layers, in fact, okay? So when we do this type of operation, it's called a lossy compression, you lose information when you do this, okay? So sometimes if your data is your data set has many wavelengths, sometimes it helps to compress because you have a lot of collinearity between the wavelengths. Okay. Personal experience, most of the times you lose information and your final predictions get worse. Yeah, this is just personal stuff. So you can use also batch normalization. Usually layers, it stabilizes the training process. So after each convolution, you do a batch normalization because usually data is fed into the neural network by batches. You can do batch normalization. And this batch normalization, will be also important, I guess, in the future, because now we are seeing something called in-context learning that allows the model to learn stuff as it looks to new samples. And one of the mechanisms that the model can do this in real time, adapt to new data, is by retraining the normalization layers in your model with the statistics from the samples you're predicting. So in some cases, this leads to models to perform better on newly data just by doing this batch normalization on the fly, okay? But this is very experimental. I have not made it work yet for the case of 1D spectra, but I know that people have done it for 2D images and other type of data modalities. We can use dropout layers and regularization to decrease overfitting. Actually, if you talk to computer vision people that do hardcore deep learning with very deep models, they will tell you that overfitting is no problem. Regularization is so high that models always behave well. In very deep models, it's very counterintuitive. You have millions of parameters, but it's very, due to this regularization and penalties that is imposed on each weight, it's very difficult to overfit, okay? So usually we overfit when we are careless or we don't have the architecture well planned. So that's, I think we, and I'm talking about me, that don't have an in-depth knowledge of this type of computer science hardcore background tend to do some errors. We can have dense layers, another piece of the puzzle. We can have skip connections. So instead of going to one layer, it skips from one layer, it skips a layer and goes directly to the other. For example, here we can have a skip connection from, imagine that you have two convolutional layers. The data can go from pass to the first convolution and go there, and then pass directly to the second convolution and be there. Sometimes when you have problems with gradient descent, so when you backpropagate your error from the, so you pass forward all the weights, do the final calculation, calculate the difference between your real measurement and your prediction, you have an error, then you backpropagate the error through this, and when you backpropagate the error, you change the weights a little bit, right? And then when you arrive to the first layers, if the weights don't change that much because your network cannot learn, then you start to have collapse of your model, okay? So all these techniques help a little bit with this kind of problems related to technical stuff in training the neural network. And then there's multiple architectures that may express the same function. And this is, well, this is This is why I think that sometimes, even if you don't really optimize your model, you sometimes get very satisfactory results. Because there's something in artificial neural networks called the universal approximation theorem. It basically says that a neural network, a multi-layer perceptron, a dense layer, with a certain number of units or neurons, can approximate any continuous function. Okay, as long as it is continuous, that thing can fit perfectly that function. Okay, so if you think of as a convolutional neural network or artificial neural network has a bunch of mathematical operations, matrix multiplications, etc., you can really understand that you have just two layers. You are restricted by the number of operations you can do. But you have 5 layers. Those 5 layers can mimic the same operations that you do with 2 layers, right? So the mathematical range or expressibility that you have with 2 layers, it's contained within a model with 3, within a model of 4, within a model of 5, okay? So this is something that's called network expressivity. It's called the ability of the network to actually model your data. And what we have seen so far, the couple of years that we have been working on this, is that usually small neural networks work as well as big neural networks. Okay? So there's some examples like deep spectra. They use this inception model, which is a bunch of 1D, 2D, 3D, 1D convolutions with different kernel sizes. Then everything is concatenated and has a skip connection. And this is called an inception model. It comes from Google Inception. And they use this, several of these modules. It's very regularized, so it doesn't overfit. But you can you apply this deep spectra from Charles Zhang to different data sets, and you don't have the best results. Sometimes you can just replicate or improve the results. I wouldn't say sometimes, I would say most of the times, with a new data set just by doing a good optimization of your hyperparameters of a 1D model. So how do we start if we want to create a neural network for our model? We have a data set, whatever, rice spectra, wheat spectra, and we want to, okay, I'm going to start looking at this. There's 2 paradigms at the moment. I'll start by the last one, which is to use a predefined architecture with some known inductive biases. So what do I mean by inductive biases? Inductive biases is something that I know it results from my physical intuition or expert knowledge. For example, what I was telling you before, if I have Raman spectra, I'm going to use narrow kernels, narrow filters in the first convolution. If I have NIR, which is very broad bands, then I'm going to use wider filters. This is an inductive bias because I am putting some knowledge that I think it might work directly on the architecture. Okay, it has the advantage that it has a smaller hyperparameter to optimize. Okay, it usually tends to increasingly be more complex than it should. It may miss the best architecture for your data. Okay, and so far as I can tell, my personal opinion, no one has made a architecture fitted for spectral data. Okay, if you want to go to 2D data and you want to do segmentation, then you have YOLO and Dino architectures. If you want to do text prediction, then you have transformers that work very well there. If you want to do spectral analysis, CNNs work well, but there's multiple ways of building a CNN, right? So there's no specific architecture yet that we can apply reliably to every type of spectra, and it works fine in every occasion. We are not there yet. The advantage is that we might be able to introduce some expert knowledge into these models. Now, there's the other paradigm, which is we go full hardcore. We don't know anything about our data, and so we perform a full neural architectural search and hyperparameter optimization, so NAS and HPO for each data set. Okay? So I don't know what is the best combination of layers and which hyperparameters work best for that data. So I find a way of trying 1 layer, 2 layers, 3 layers, wider filters, lower filters. And the problem here is that the combinatorial number of possibilities we have to search becomes, well, it explodes. So we need to resort to some clever search methods like genetic algorithms to do the optimization, or Bayesian optimization. That's the one I prefer. And it usually results in simpler architectures because you can penalize for complexity. And usually what you have is like, in the end, you have like 2, 3 layers, and it gives you the best result than very complicated. Also, that might happen, I have to disclose that, because as you increase the number of layers and the number of hyperparameters, right, the optimization problem in hyperparameter space becomes much more complicated. Okay, so if instead you have like this 2D plane where you have your concave optimization function and your minimum goes traveling, goes there. No, you have to think, if you have 10 different hyperparameters to optimize, like number of layers, width of the layers, each number of neurons in each layer, regularization strength, etc., then you have an object optimization in 10-dimensional space. And I guarantee you, your physical intuition beyond 3 dimensions doesn't exist unless you're a mathematician. Okay, and then you have to get inspiration to do these kind of things. But that's the problem. Okay, so our physical intuition for optimization problems beyond 3 dimension, it's very poor. So this is an example of an heuristic bias that sometimes works with image and sometimes works with spectra as well, but not always. And that's the problem. We do a bunch of things, and sometimes it works, sometimes it doesn't. It's difficult to find the patterns. And it is like, for example, you have 3 convolutional layers, and as you go deeper, you decrease the number of the width of your kernel, okay? So for the first layer, you have 7, so it means that this unit here sees 7 wavelengths, and then you decrease the thing, but the receptive field, the amount of information that the pixel in the 3 convolutional layer sees, it sees much more information from more wavelengths. And if you go 1 level deeper, the receptive field increases. So as you go deep, this unit here can have information about a large part of your spectrum. Okay? And if you go deeper or you change the widths of these things, you can have combinations. For example, we can have multiple filters here. A filter, one of the filters detects increasing slopes. One of the filters detects decreasing slopes. And one detects inflections, for example. Okay? Then when you combine them, in the second layer, you have an increasing and decreasing, and then a slope. Then you start to detect peaks. Okay? And then when you go here and you see all the information from that part of the spectrum, you start to, one peak here, they always change in shift. They shift in phase, so they might be related. And all the way here, okay? So low-level features, combination of these low-level features into high-level patterns in your spectrum. This is what it is supposedly doing. Is it doing it? We don't know, because there's a bunch of variability in the data that we usually work. right? I'm going to tell about the type of data that I work. I don't know if it has, looking backwards now, it has not been the wisest choice for sure, because I have been working with food data, mainly fruit data, and we have different seasons, different temperatures. Sometimes the spectra are collected in the orchards, sometimes the spectra are collected in the lab, and there's a bunch of variability factors that affect how my spectrum behaves, that when I try to make this mental picture, oh, this pixel is changing because of the amount of sugars. No, it's the amount of sugars plus the position of the spectrometer plus the illumination fluctuations, all of this. So what we actually need to make progress in this part, this is a tip for you guys if you want to do, it's try to come up with a standardized, very well behaved spectral data set where you know where are the sources of variability inside that spectrum. It can be synthetic, it can be very high quality spectra taken in the lab, for example, for exactly same temperature, same spectrometer, but very precise amounts of, for example, I don't know, drops of sugar inside water, increasing the concentration. Do this, a large data set, and trying to pick a very well behaved variability source from your spectra, and then model it back and see if you can regress it, okay? So as I was saying, this is related to the early layers, basic filters, narrow layers, and deeper layers go to combinations. Now, if we only use one layer with one filter, this is something that I like to do, you don't have feature combination. So we cannot talk about hierarchical learning in this sense. What we're doing is learning a pre-processing, because with just one filter, in one layer, we're learning a small window that does something that enhances the capability of these certain features to be to contribute more positively to the prediction. So this is one of the things that I find in the literature, is that sometimes people use preprocessing, sometimes they don't need to use preprocessing and they get good results. Well, I can skip to this part straight away. I think that in most cases, preprocessing is needed when your model is simple and not well optimized. Okay? I cannot think of a way why if you do it with proper optimization, for example, if you have to linearize your spectrum, like a 1D or 2D derivative, in order for PLS to work well with it, I don't see any technical reason why the CNN shouldn't be able to learn that. Okay? It's just a set of weights in a sliding window, and that is what's supposed to be learned by the CNN to make the prediction, of course. So I guess that most the varying results between, oh, we have to use preprocessed data, well, we don't have to use preprocessed data, it comes from the poor optimization of the models, okay? In person, I found that when you use the preprocessing that usually works well for PLS, since we used very simple models in CNNs, they tend to be linear plus 1 or 2 orders of approximation afterwards, they work well as well. So if you have, my advice is that if you have done your work for PLS and you want to test your CNN on it, feed it the preprocessed data first and see if it works, okay? Usually we have to standardize the spectra before feeding it to the CNN due to the statistical properties of the weights of initialization and how gradient descent works, okay? So standardizing the data, it usually helps because many of the algorithms used and the activation functions assume a zero mean-centered data, okay? So it helps a lot. So how do you start using CNNs? First, you do what Tom said, okay, yesterday. You correct your data partition, you do the preprocessing, you do the PLS first or local, whatever, and if it works, good, you're done. You don't need to go to CNN, okay? If you're not happy, well, welcome to the house of pain. You start with a shallow CNN, okay? One conv layer, one dense layer, and you play with the hyperparameters to get a feeling if you get the range of values that you're supposed to have for your prediction. And then you go a little bit, if you, through trial and error, you didn't find anything that suits your modeling needs, you proceed. Now you go to another step, which is automatic hyperparameter optimization. Just the hyperparameters, keeping the architecture fixed. And you either use a random grid search or more computationally efficient Bayesian methods, and you optimize the crap after those hyperparameters. If you do it well, then your model will start to behave well. If you need to go deeper, then you start to play around with neural architectural search. Either by trial and error, you try different combinations of layers, or you can, as we will see in the tutorial, there is a couple of ways, you can just do optimization of the neural architecture and the hyperparameters just in one go. If you decide to go that, you put your process running and you go do something else. Because this usually it's very time consuming, okay? So it's very difficult if you don't have access to the computational power, okay? Or if you have to do this for research and you have to do it multiple times. And then you have to, well, you analyze your data in your tuning set or your validation or cross-validation set performance. You choose the best hyperparameters based on this metrics, and then you compute the test set. Sometimes what happens, because you have different distributions in the data, it's what happens with every modeling. Your validation set is quite nice, and then you go to predict the test set, and so, and then you repeat, and then you go do something else again. So now we're coming back to interpretability. So interpretability and explainability, there are two different concepts, okay? Interpretability means that I want to understand what took or what kind of features did my model use to make the prediction it did, okay? Explainability means understanding how it did it, okay? So for interpretability, we're lucky because people from machine learning have devised techniques like LIME, SHAP, and regression coefficients, where we can apply this to any type of model, black box or not, and we can figure out what the model uses to make the predictions, or what the model pays more attention to make the predictions. So there's this book, it's online, by Christoph Molnar. It's very good if you want to have an introduction. It also has code if you want to implement it on your stuff. And there's a couple of reviews here that also show you a bunch of methods that might be helpful. So just to come to tell a little bit about one of the examples that we're going to talk about, it's called Shapley additive explanations. And this is something that was developed initially for for game theory, okay? And what basically does is you have a team, and this team has an objective, and you want to compute what importance does player A, B, or C, what is the contribution of player A, B, or C on this team for the team to have this objective. So basically it's a bunch of permutations, okay, that try to see what is the value of that wavelength, let's call it wavelength or wavelength band, because we're dealing with spectra, for the calculation of the thing I want to regress. So there's a bunch of algorithms that do this feasible, because this is when the number of features becomes high, it becomes highly intractable if you're going to do all. But there's a bunch of tricks, technical tricks that can do this. So SHAP values also have a nice property that I like, because it's that if you have your model and your model, you can compute the average prediction of your model. The interpretation of the SHAP value for a certain feature is how much above or below the mean prediction that feature contributed. Okay? So for example, if I'm trying to predict moisture, and we have a peak at 907 nanometers due to water, and I have an average value of moisture, and I can almost certainly be sure that features around 970 is going to have a positive weight, a positive Shapley value. They are going to contribute to increasing the amount of the number that I want to predict. So there's an easier interpretation of Shapley values. Well, why do we need to go this way? Well, it demystifies a little bit learning, because it relates to features in the chemistry, and for many people this is important. It's a diagnostic tool to refine models, because if we see that there is some bands that are always contributing to mess up my results, we can filter them. It enables deep learning plus domain expertise models, if we do this, including a filter, for example, in the beginning. But there are some hurdles. Well, model complexity hinders chemical interpretation. If your model is very complex, it does a lot of things to your input data. When you try to unfold this, it's going to be very difficult, right? And these algorithms, what they try to do is try to unfold what has been done. So model complexity for certain points, it's more difficult to make them transparent at all. If you actually need to go beyond this SHAP analysis or coefficients like this agnostic models, and you want to use other stuff like kernel, what is the shape of your kernel, what does your convolutional layer does to your input signal, then, well, welcome to Team Deep Learn Chemometrics. This is one of the things that can also be used. It's called GradCam. People have already mentioned. So basically, you have an image here. It also works for spectra. You have an image, you have a convolutional layer that does some classification. And depending on the classification, if it is like a dog or a cat, we can backtrace the gradient and see where the weights come from. Okay? And then you can have these saliency maps that show that, for example, red, it's a dog, and green, it's a cat. So these can be helpful in some contexts to know if the spectral band is related to what. Okay? Which spectral band contributed to my prediction. So we have to be very careful with results reproducibility. Okay? There's a bunch of things in technical deep learning that can make your results fluctuate a lot. One of the things, it's if you're running on the CPU and every matrix multiplication is sequential, fast but sequential. Or if you do on a GPU, when you do a bunch of them in parallel, but then there is a problem with recurrence, because if one matrix multiplication finish first, then the other, due to some reason, some pipeline, some delay inside the, you're going to have different truncations in the algorithms in the 6th or 7th decimal house. And then when you aggregate all of that, you get variability. Nowadays, there are some methods inside GPUs that try to combat this randomness due to parallelism, but okay, you have to be aware of them. And then there's the one of the biggest things. It is, you have to learn one of these frameworks. TensorFlow, JAX, PyTorch, okay? This means that you have to become a little bit a programmer. If you have a background in MATLAB, you can do it in MATLAB as well. It's not as flexible, but you can do it. One of the things that hinders the adoption of this is that this ecosystem, due to this deep learning hype, it's skyrocketing. It's evolving every time. I do a model. I spend like 3 months or 4 months working on this. If I do upgrade my software stack, it will not work because now it has a new feature. It changed the dimensionals of... So this is a nightmare. Be aware. Okay, how much time do I have? Sorry. Is anyone keeping track?
Well, it's already supposed to be finished, but we started late.
Okay. Give me 5 minutes and I'll just fly here. This is just an example that we did a couple of, like last year and this year. We call it Deep Tutti Frutti because basically it's a data set with different fruits, okay, all taken with the same spectrometer, the Felix F750, different apparatus, but the same model, okay? And we use the second derivative preprocessed. Why do we use the second derivative? Well, because the original data, one or two data sets were recorded directly using the second derivative export from the from the spectrometer. So we don't have access to the raw data. And if you want this data, you have to talk with Puneet Mishra. He has authorship of the data. It was available online, but is not anymore. Yeah, we have to talk to Puneet. So the data set is apples, pears, mango, and kiwi. And when you look at the second derivative, this is what you see. This is biological stuff in NIR, and we're trying to predict dry matter content. Okay? And you can see that at the naked eye, you cannot distinguish the spectra. They have the same shape in the second derivative as they're supposed to. But when you look into like this kind of dimensional reduction techniques, this is like a sort of non-linear, more squeezable PCA. Okay? You can find pretty much, if you fit in the spectra into these T-SNE embeddings, you can see that they separate, and even better, even the same type of spectrum, for example, here for mango, you can see that there are at least 4 clusters. And when I went backtrace the origin, yes, you have 4 different batches. And the same thing for the pears, you have actually 2 different batches coming from 2 different places. So this is nice to see. It's nice to see that we can use at least this to get some information about the subsampling of our group. And then what we did is something that we'll do in the tutorial. We have the full data set, we do a split, then we do some hyperparameter optimization using Bayesian optimization. Okay? We have an optimized CNN, then we fit in the training set into the optimized hyperparameters, we get an optimized CNN, and then we predict the test set in the end. Okay? We also did it for multiple fruits, but we also trained on multiple fruits and tried to predict single fruits. Okay? And we also tried to predict single fruits from another data set that was not in the training set. And it worked satisfactorily well, better than alternative methods. So why does it work well? Still learning. So this part here, the cross-validation strategy is something that you can do for optimizing your model. We did a couple of different things, like 5-fold cross-validation. So mix every type of fruit together, split it in 5, do the cross-validation. Then we also did cross-fruit validation, like choose 3 fruits, put the spectra for training, and try to predict the other fruit. Okay? This was the model that performed the worst, okay? But still below 1% of dry matter prediction. So most around PLS. okay, even the worst performing. So these different ways of optimizing generated different architectures, and we're going to see these architectures. They're available here. There's the link. You're going to have the slides, so you don't need to take a look. There, we're going to share the PDF of the slides, so if you want to take it, copy it then first. The code done to do this, it's available. You can make it, you just can copy it from my GitHub. And you can try to look into methods to compare and understand where does CNN work. In this case, what we did was we trained a model with 100, 200, actually it's intervals of 50, I think, or 100 maybe, I don't remember. So 100 and 100 samples in the training set, and then predict the test set. Training with 100, predict the test set. Training with 200, predicting the test set. And we see that while PLS, after just 200 samples, it stabilizes the performance. Okay? And this is a properly optimized PLS because it's optimized the preprocessing for each data partition. Okay? So after 300 samples, CNN starts to still learn and improve. 500, 600, 700, all the way to maybe here. And then it flattens out. So if you get this type of result, what do you do next to improve your model? Usually people say, oh, get more data. It doesn't matter. The CNN we have, you can feed it more data, we will stabilize there. Because why? Because these models are simple, and the expressivity of the network is not enough to capture any more information. Okay? This is just a couple of layers. One convolutional layer, one dense layer, one or two filters. So in this case, if I would continue this, I would try to increase the number of layers, for example, or the number of filters, or the number of neurons in the last layer, and see if it is changed at some point. If it goes a little bit lower, if it goes faster here or not. Okay? So this is one of the things you can do to start interpreting how your neural network actually works with the data. Then we also computed the SHAP values. So for SHAP values, we did the same thing for one of these CNNs, and it gives me information about what wavelengths contribute the most to the prediction of dry matter. Since it is a model agnostic, we can do it for PLS. It's just a box. It doesn't matter if it's transparent or black. Okay? You compute SHAP for the value for PLS model. This is the black line. And you do it for the CNN. And you can see that CNN pays attention to a couple of different bands that PLS doesn't care of. Okay? But the main contributors are these ones here. And the same thing for the VIP score, the same bands are identified. So this reassures that PLS, and that we understand and love and trust, give more or less the same results as our CNN. This lets us give some confidence that our CNN is not just stochastically combining stuff. It's picking up information from the relevant bands. Okay. Now we can also use GradCam and do the backprop and see what bands matter the most to identify each of the fruits. And remember, we standardize the data in the beginning. There's a bunch of apples and then a few other fruits. So when we standardize the data, we use the mean of the whole data set and the standard deviation of the whole data set to standardize. What the standardization is going to do, it's going to enhance the differences between the spectra of apples, mangoes, pears. So that's why we are able to see that different bands in different fruits contribute differently to the prediction of not dark matter, wrong paper, to dry matter. Okay? So, yeah, I'm going to skip this one. just telling you that, for example, for cross-fruit validation, this is the CNN. It's the same CNN, okay? Just the validation used to optimize was different. This is cross-fruit validation, so you optimize, you train on 3, predict the other in the validation set. And here you do cross-validation, do this 5 times, okay? In this case, the filter that was found has 3 units. So it's a very narrow filter, okay? Why does this happen? Speculative, my opinion. Because when we were trained on this fruit, there are no large patterns with the same type of variability that this fruit has. So the neural network tries to find small features, okay, that correlate between this and this. Now, when you have everything mixed up, you want to predict everything mixed up, so the kernel goes out to 35. You try to get a large portion of your input, and then you slide it to get these patterns, okay? Then you can go a step forward, and you can look into what goes into the neural network, which is the original scaled spectra, which is the, can't see the color, sorry. The black one, this is what goes into, and this is what comes out the convolution. So what do you have? You have small peaks here in these big transitions, okay? Small peak here in this transition, small peak here in this transition, and then smooth. So what did this filter did to the data is smoothed it out and highlighted the big transitions. Do you recognize this type of form? This is like the coefficients for a second derivative. Usually for second derivative, you have 1, minus 2, and 2. And here you have 0.5, minus 0.5, 0. So it's doing some baseline shift and enhancing these kind of things. Now then you can go to the other side and see that there's more complicated filters. What is this doing? I don't know. It might be a combination of other type of filtering, okay? And this is just one filter in one convolutional layer. And this is what the neural network found that worked best for this type of data. If you go to 3 convolutional layers with multiple filters per layer, what you see here in the first layer, it's, for example, it found 27 filters. It works best, wide filters. This is the convolutional, using the strategy of 5-fold cross-validation. So train on multiple fruits, predict multiple fruits. So you can, again, get this wide filters. This is something that I see recurrently in biological data. When we do optimization, we tend to get wide filters. And here you see like a bunch of null filters. Those are non-responsive, have almost flat 0 response. They don't influence. This is something due to redundancy in the neural network or lack of data to explore this kind of thing. But then you see a bunch of these wiggly waves that behave like frequency detectors, like we see in patterns in signal analysis. So it detects certain frequencies. Some of these can be related, for example, like do the same kind of operations that what's called the Dirichlet. It has this hat. How do you call it? Wavelet. The kernels that wavelets use to filter data. These behave more or less the same thing. Then here starts in the second layer. You combine all these things and start to emerge more complicated patterns. And in the third layer, you have already some peak detections, some patterns that can be or not, I don't know because I didn't went that far, related to the initial spectrum. Remember that these filters end up seeing, because the receptive field is going to increase as you go deep, they end to see the whole spectrum, okay? So this convolutional filter interpretation has been done also by other people. They can relate it to some vibrational bands. It has been done in the past as well, so it was not just that result that I showed you. And well, just to finish, last slide. Even though most of the works in this area do not properly optimize the model, because they don't properly optimize the CNNs, they do comparisons between CNN and something else, and they say, oh, this model is better than CNNs. The CNN was not optimized. It's like the same thing in the beginning with PLS. PLS doesn't work here, regression works better because the PLS was not optimized. Okay? People don't tend to actually put the work into optimizing the CNNs. So most of the comparisons you see, if the optimization is not done properly, it's misleading, my opinion. Okay? Most CNNs are shallow. Okay? What we're doing is what AlexNet did 20 years ago in computer vision. So our models are baby stuff for computer scientists. Most computer scientists, these guys, have never learned about NIR spectra. They've never seen that is a data modality nice to develop deep learning models. So that's why you have just for image, text, sound, but NIR spectra is a data modality that is unknown to these guys, okay? And we should try to make collaborations with them, bring more computer scientists into the field, okay? CNNs can be interpreted if your objective is to say, I want to make sure that my model is taking the information from the right spectral bands that we know from chemistry. You can do that. It's not a black box in that sense. Okay? It's like VIP scores. You can do it. Reproducibility requires proper handling. I've told you this. And deep learning chemometric models are not, and I don't think it will ever be, a set and forget. This is the holy grail. You establish your chemometric model, and then it works forever. I don't know if even deep learning will be able to do that, to be honest, at this point in time. Well, CNNs are resilient to overfitting, and I have to stress this out. There are some wacky examples in the literature of deep learning. You have really nice phenomena that occur, like double descent or grokking. These are things like you over-parameterize your neural network and you expect it for overfit like hell, and you train it for a bunch of epochs, and what you see is that the performance flattens out on the training set. It doesn't learn anything on the validation set, and after many decades after the training, it poof. So even an overly parameterized model with millions of parameters, and this is why LLMs work as they work fine as well. If you take proper care of the regularization, you don't overfit your data. Okay? And well, nowadays, this is still in infancy. There's in the same way, a couple of years ago, when you did a PLS, the validation number of latent variables that minimizes the validation set is 5. Now, but I'm going to choose 4 because I know it generalizes a little bit better. It was like in the beginning when I started, it was like I look at the elbow and it's more or less here. Why? There is no specific criteria. There was a little bit of black magic. Believe me, CNNs require a lot of more black magic, dark magic in the moment to craft than the PLS. Okay? So if you really want to get into this and you want to do, and we'll start to see a couple of examples, you pick up a book like this. This is from a researcher, Simone Scarpadane. So anyone here from La Sapienza in Italy? No, yeah, it's a professor there. Machine learning guy, very nice book. He has all the theoretical stuff written in for non-technical computer science guys, like for physicists and chemists, so it's very good. You pick up a model online, you pick up your wars, your arms of war, like your Python, your MATLAB, or whatever, and you start coding. There's no other way at the moment. For PLS, super good. You install MATLAB or eigenvector, whatever, and then you have the PLS toolbox, and you go there and click, 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 and so nice. If you need to do that for a project, yes, do it. If you go into CNNs, do it properly, and prepare yourself for a world of pain. Okay? So this is not to discourage you. Okay? It's just to put things on the table as they are. CNNs at the moment work well, but they are not practical for most people because it's not a very clicky, clicky, clicky thing and automatic to do. We're still not there. But if you guys want to do research on this, welcome and thank you.